Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Robert Vassar, the director of the Mesulam Center for Cognitive Neurology and Alzheimer's disease at Northwestern University. We will be discussing the future of neuro research and the hope it brings. So let's dive in. I would like to just kind of start with, how did you get into research?
Well, I always loved science, even as a little kid, was my favorite subject. I actually wanted to be an astronaut when I was little. I didn't quite make it, for a couple of reasons. First of all, I wear glasses, and you have to have 20/20 vision, back then at least, to be an astronaut. I was also a overweight kid, so I couldn't, fitting into the capsule might have been a problem. And I had claustrophobia, so
yeah, that's not going to work out too well.
That wasn't going to work out. But, my great aunt, my great uncle was an MD, and my great aunt really kind of pushed me to become a pre-med. And I started at the University of Chicago as a pre-med. But then I realized, I really enjoy biology more than I do medicine, because back then it was a lot about memorization and not really understanding the underlying science of it. And so I shifted to biology. And after I graduated with my BA, I didn't really know what I wanted to do yet. So I spent some time as a technician in one of the labs at the University of Chicago. And that was really a nice period of time for me to allow me to gather some technical skills and just kind of think about what I wanted to do. And then in 1983, my mother was diagnosed with Alzheimer's disease. And then everything fell into place. It sort of crystallized for me what my purpose in life is, and that's to study this disease, to try to understand more about it, to figure out what makes it tick so that we can stop it. And that kind of set me off on a kind of serpentine voyage, which started with graduate school at University of Chicago, first in neurobiology, but back then, in 1986, there wasn't much known about Alzheimer's disease. The genes were not discovered yet. The pathology was known, but nobody really, understood much about how it was formed. And so I ended up taking a side road to learn technology. Technology, in this case, was transgenic mouse technology, which was not available at University of Chicago at the time. So my PI, Elaine Fuchs, sent me to another lab to learn transgenic technology and bring it back to the lab. And then I used it to study the skin. Eventually, after graduate school, I got back into neuroscience, working in a lab at Columbia that studied olfaction. And this was Richard Axel's lab. He ended up later winning the Nobel Prize, partially for the work that I was doing in his lab. And that was a real experience because Richard taught me how to think. I had some good technology under my belt, technically, in terms of skill set, but watching how he approached a scientific problem really helped me. A light went on. I said, oh, I know how he thinks now. And I'm going to use that same approach to my science. And that's been invaluable for me. after my postdoc, I joined a company, Amgen, to do my first Alzheimer research at that point. And, we made some great discoveries. We discovered an enzyme called BACE, beta site APP cleaving enzyme, that is involved in generating the beta amyloid that forms the amyloid plaques in the brain of Alzheimer patients. And BACE is a beautiful drug target. There have been drugs that have been developed, but they've been put on pause because of side effect issues. But some colleagues of mine and me, we're working out new clinical trials to try to avoid those side effects. So it's still, a possible therapy in the future for Alzheimer's disease.
Yeah, that boy, from being an astronaut to studying the olfactory bulb to this transgenic mouse technology. What is that?
Well, it's a form of genetic engineering that we use in mice. Mice are great model systems for disease and for biology. They're like little test tubes. And back in the 1980s, the transgenic technology was developed to alter their genomes in such a way that they express, a gene of choice, whatever gene you want. You could put it into the mouse and have it express that gene. If you wanted a gene, a normal gene, you could express that. If you wanted to express a disease gene, you could put the disease gene in. And in fact, at Northwestern, I've made an Alzheimer's disease transgenic mouse that develops the amyloid in the brain. And it's actually one of the most well-used mouse models for amyloid pathology that's in the field. And so these transgenic mice can be very, very useful for understanding human disease and the actual pathogenesis of disease, because obviously in people you can't do the sorts of experiments that we can do in mice. Although things have gotten technologically a lot better in humans to be able to follow the pathology in a living person using, for example, positron emission tomography and blood biomarkers to watch the formation of these pathologies in the brain. But still, when you want to get to the molecular and genetic level, the mouse is really the best.
Yeah, that's super interesting. So you had mentioned that you understood this type of thinking, like how to think, when you were studying the olfactory bulb and that function. If you could describe what that type of thinking was that you just really just kind of clicked, and then, how do you bring that to what you do today and to those people that you're mentoring?
Yeah, this was really eye-opening for me too, when I had meetings with Richard Axel as a postdoc. And we would sit in his office. Richard had a big couch, and he would just lounge on the big couch. I'd be in a chair across from him. And, we'd be talking about my science. And I would be explaining to him, my thoughts about the experiment and the experimental results. And he would stop me. He said, "Now hold on, let's back up." And he would then methodically go step by step by step, almost in baby steps, through the logic of the experiment and the results. And I realized what I was doing, and I think it's really common for us to do this, is I would just kind of leapfrog over the data and not really examine it in a rigorous step-by-step logical fashion. And I think that's been one reason why many of our clinical trials don't work. And especially in the Alzheimer field, we've had up until recently, a really bad run of, 99% failure. But recently we've had a couple successes, and we still need to do better, but it's really hope on the horizon there with these successes. But this methodical thinking that Richard brought to bear and that I learned from him was extremely valuable for me as I've gone through my scientific career.
Yeah. Well, thank you for sharing the story about your mother too. That that influence and that that kind of drive that's pretty deep seated. How was it? You said she was diagnosed in 1983. How is the diagnosis process, diagnosing this disease? How has that changed from 1983 to 2024?
Wow, it's been vast. Back then in 1983, all we had were simple memory tests to be able to diagnose someone with memory impairment. And even so, back then, we could never say for sure whether a person had Alzheimer's disease. We could just say, based on their memory test, that they had impaired memory and cognition, and that they were exhibiting a form of dementia that was probable Alzheimer's disease. And you really couldn't have a definitive diagnosis of Alzheimer's disease until death and autopsy and a neuropathological examination of the brain to look for the hallmark lesions of Alzheimer's in the brain. So that was 1983. And today what we have are a whole slew of different technologies that we can bring to bear to diagnose people with Alzheimer's disease. The first was a cerebral spinal fluid measurement of certain what we call biomarkers in the CSF. Next came brain imaging. We have MRI to be able to look at the volume of the brain, and there's significant brain atrophy during Alzheimer's disease, especially in a region of the brain called the hippocampus, which is important for memory. And so seeing, atrophy of the hippocampus is one more sign that the person may have Alzheimer's disease. Then came along positron emission tomography with the radioligands that light up the amyloid pathology and also the tau pathology now. So we have these two measures, these two brain scans that allow us to actually see the pathology building up in the brain, even before there are memory symptoms, which is exciting for the future because I think that these new drugs that have been approved may be useful for prevention of Alzheimer's disease if we give them to individuals that have evidence of the pathology but don't yet have symptoms. And in fact, there are a few clinical trials going on right now for prevention with these drugs. So we'll see what the future brings. And maybe most excitingly now, we have blood tests to detect these pathologies in the brain. And they're not yet FDA approved, but they're getting very close. And that's very exciting because in the future, I see us, going to our primary care physician, just like we get blood tests for cholesterol and other vitals in our body, we'll be able to have a blood test for Alzheimer's disease to see whether the amyloid is building up in the brain or the tau pathology. And that's going to be a game changer because when we have more drugs that block these pathologies from occurring, then we'll be able to treat people before there's damage to the brain and memory impairment. That's huge. Then I think we'll have a true prevention for Alzheimer's. Prevention is going to be the key because, and it's true for all diseases, especially diseases of aging, like heart disease, diabetes, and even cancer. If we can take measures to prevent these diseases, we essentially cure them, or at least we delay their onset to the point where, it's not an issue anymore. So,
hey, can you imagine if you start when you're 22, 23, whatever that age is, and you start getting these routine blood tests in your annual physicals, and all of a sudden now you can start to see the change over time. not wait till it's too late. That's pretty amazing. And I appreciate you stepping through kind of these big stages of technology. What's on the horizon? What's out there in the next 5 or 10 years on the technology? What do you see as the big step there?
Well, I think, first of all, the new drug targets that are being developed right now and therapies that address those targets, the anti-A beta immunotherapies that have been approved recently are the first step. They show real disease modification. In other words, so these clinical trials were in people that had Alzheimer's disease. So they already had the memory deficits, the memory problems. So those drugs did not stop the progression, but they slowed it down significantly. And that is a true measure of disease modification. That means we're blocking the underlying biological effects of the disease process, which is huge. So amyloid is what we think is going on is that it in and of itself is probably not super toxic to the brain cells, but it starts a process that is kicking off another pathology called the tau pathology, which is the really bad player in Alzheimer's disease. So amyloid triggers the tau pathology. Now, what we need to understand more about is what is the connection, the mechanistic connection between amyloid and tau. What is it about amyloid that kicks off the tau pathology? And my lab here at Northwestern is really focused on that question. It's a profound mystery right now. And we're excited that we're making some baby steps in that direction. We know that the tau pathology is bad, and there are therapies under development right now, tau immunotherapy. So these are antibodies, just like the A beta antibodies, but those antibodies are directed against tau. And so the thought there is that administration of these tau antibodies may clear out the pathology, the tau pathology, or prevent it from spreading. The other new technology, which is really exciting, is called antisense oligonucleotides. Or I'm going to abbreviate them ASO.
Yes, please.
These ASOs have been proven in other diseases to work. And there are a few examples of ASOs working for other diseases. So now they're being applied to Alzheimer's disease, in particular, ASOs directed against the tau pathology. So the idea there is that it's really a gene therapy approach where the ASO lowers the expression of the tau. And that's been shown actually in some limited human trials to slow down the buildup of the tau pathology in the brain. Now, these are very early results. The number of patients is very low, but it looked real to me when I saw it. It was presented a couple years ago at the Alzheimer and Parkinson's disease meeting in, I believe it was Lisbon. And so we haven't, these trials are very long. We haven't seen any more data since then. But I'm very hopeful for that new technology. Also, there's a lot of inflammation in the brain that these pathologies cause. And there's a cell in the brain called the microglial cell. And microglia seem to be, we're not quite sure whether they're good guys or bad guys. I think they're very complicated, like people. Every person has a yin and the yang, right? They have a good side and maybe a not so good side. Well, the microglia kind of can change. They're shapeshifters. They can change from a good microglia to a bad one and vice versa. And so we need to understand more about what makes them tick so that we can kind of help them become good players in the brain and not bad players. And so I think there are many approaches, therapeutic approaches in that area as well. So there's a lot of, therapeutically, there's really a lot of new exciting things going on. Technology is always the driver of advancement in science. We talk about omics these days, genomics, transcriptomics, proteomics, lipidomics, you name it. And it's kind of a unbiased approaches to look at changes in the brain that are associated with Alzheimer's disease and how those changes progress as the disease progresses. And we're learning a lot about the disease process through these omics. Another technology is human-induced pluripotent stem cells. And so, I'm a mouse modeler. I use mice a lot, but obviously there are differences between the mouse and the human. And sometimes those differences are really important. So now we have these, we call them iPSCs. These iPSC neurons that are human can be grown in a dish, and they're beginning to be modeled, or at least they're beginning to model diseases like Alzheimer's disease, Parkinson's disease. And, I think what's even more exciting is that we're assembling these cells, the iPSC neurons, with iPSC microglia and astrocytes to form what's called an organoid. It's like a mini brain in a dish. And so what we're really hopeful there is that we can model a tiny little human brain in a dish and see how the Alzheimer's pathology progresses in that model. And we're still not there yet. We have a long way to go to develop this technology. But I think there are going to be a lot of interesting questions to ask and answers in the future using that approach.
Yeah, it just seems like it's moving much faster than it did 40 years ago when you kind of started on your journey here. You're the director of the Mesulam Center. I hope I'm pronouncing that correctly. Mesulam for cognitive neurology and Alzheimer's disease. Tell us about that center, because you've talked about your lab. Is that the center is your lab?
Oh no, the center is much bigger than just my lab.
Okay, so talk about the center, talk about how your lab fits in, and just what is kind of the day in the life of Dr. Vassar.
Wow. Okay. Well, let me start out by talking a little bit about the Mesulam Center. So Dr. Marcel Mesulam started the center 30 years ago. And he's a cognitive neurologist that discovered a type of dementia called primary progressive aphasia, which causes problems in speech and language. And it's a very interesting dementia because it's different pathologies can cause it. Even Alzheimer pathology can cause primary progressive aphasia. I'll just abbreviate it, PPA. And so when Alzheimer pathology lands in the language center of the brain, it causes PPA. There are other types of pathologies too that land in the language center. One of them is called TDP-43, which I won't tell you the name because I'll butcher it, but it's a different type of pathology. And it can land in different parts of the brain. When it lands in the language part, it causes PPA. And there are also genetic forms of PPA. And so Dr. Mesulam established the Center for the study of PPA as well as other types of dementias, which there's a whole slew of them caused by different pathologies with different clinical manifestations. Alzheimer's disease was also in the mix there that has been studied in the Mesulam Center. But we tend to study these more rare dementias. And so, in a sense, we're kind of a boutique center. The other thing that we do is study superior cognition, which we call superaging. And that's a really exciting area of research that we do. So Our participants, our superager participants, are people over the age of 80, but who have memory function equivalent to someone maybe 30 years younger. And so, we've built up a cohort of these superagers. We also ask all of our participants to donate their brains after they pass away. And so what we can do is called clinico-pathological correlation. So what we do is correlate the brain anatomy and the brain pathology with the brain imaging like MRI and PET scans all the way to the clinical phenotypes, the clinical syndrome that the person had. So, we can make the connection. In the case of the superagers, we want to find out, how are they avoiding these dementias? And, is it their genes? Is it their lifestyle? Education? What is it? And I can probably tell you it's unlikely to be lifestyle, because these folks, they drink, they smoke, they eat whatever they want. One of their main characteristics is that they're highly social. They really love social engagement, and they're very actively involved in their families and in their friends, and they have a positive attitude. I think that is also a characteristic of superagers.
That is really interesting.
Yeah.
Yeah. And so there's no kind of common lifestyle, economic, any kind of genetic, at least right now, you don't see it. It's really about being social, which is interesting with the technology that we have right now. It's almost antisocial.
Oh, yeah.
And so, are you studying kind of the convergence or the divergence of kind of behavior in that?
Well, that's a good question. We're not quite doing that, but it's an interesting hypothesis so what you're hypothesizing is that folks that are, do more of the social media stuff rather than personal interactions might have a higher risk of dementia?
Could be.
That's an interesting question.
Yeah, because, you think about the generations that are behind you and I. I mean, we grew up as kids, and you'd go outside and play, and, you network, and you keep in contact with people on the phone or Christmas cards or whatever it might be. Today, people have thousands of friends, but they don't know who these people are. And
and it's so remote. And there's a distance there. It's not a personal connection as much.
Yeah. Because I wonder about that a lot, is maybe not our individual behavior, but our societal behavior. And because it's a very different time in 2024, will be in 2023 than it was in 1970. Are some of these social changes that we're going through, is this causing the rise of some of these diseases?
Oh, that's a very fascinating question. And I'm sure that there are groups studying this. We're not really doing it at the Mesulam Center, but we are very, very much interested in social determinants of health. That's a big, big push, also by the National Institute of Health, and we're mandated to do this work, and we are, we have cohorts of African Americans who are superagers, and so, they economically are not as wealthy as some of our Caucasian people here on the more further north. But so I don't, I would have to say probably economic factors are involved, diet, your access to health care, that is going to influence your risk. But it's not the only thing. I think genetics plays a big role, but we still don't know what are the genes involved that really, make a superager. And that's
So what
also
about your
fascinating.
lab? You talked about the center and kind of its focus. What is your lab, and what are the one or two priority questions you're trying to answer?
Well, that's a great question. So first, I am I guess you could say I've worked in a field called amyloid, the amyloid hypothesis, and it stemmed from our discovery when I was at Amgen of beta site APP cleaving enzyme, because that is an enzyme that's necessary for the production of A beta, the beta amyloid that forms the amyloid plaques. And so a big push of my lab has been to try to understand what this enzyme base does, because it's a drug target. And there are drugs that block it that have been put through clinical trial, but there were side effects. And so we're trying to understand, what is it about that enzyme that causes the side effect when you block it? And so we're investigating the different biological pathways that this enzyme regulates, not only the production of the amyloid, but other pathways that are important for the function of brain cells. So we're doing work along those lines. We're using primarily mice, and what we found out was that these drugs that block BACE, we call them BACE inhibitors. When we treat mice with the BACE inhibitors, it messes up their sleep waves. There are classical wave patterns when we're asleep, and we go through different stages of sleep. There's one stage called delta sleep, slow wave sleep. And the slow wave sleep is very important for its restorative sleep. It's important for memory consolidation. And if you don't get enough slow wave sleep, you're not going to be able to retain memories. And so these BACE inhibitors, when you give at high dose, reduce the percentage of time that the mice are in this slow wave sleep, restorative sleep. So what we're trying to do now is to see if, find a safe dose where we can avoid the reduction in the slow wave sleep. And then we can go back to humans with this data and propose new clinical trials at lower doses to see if we can get the beneficial effects of lowering the amyloid, but avoiding the side effect. So that's a big push in the lab right now. Another push that we're doing is, I said before that there's a profound mystery in the connection between the amyloid and tau pathologies. It's a black box that we don't yet understand. So we're very interested in that, what's connecting the two pathologies. We think that we have a clue in that the amyloid pathology causes damage to the membranes of the brain cells, the neurons of the brain. And the membrane of the neuron is just the outer surface of the neuron, and it's made of lipids. Now, the amyloid is it loves lipids. And so when the amyloid grows in the brain as an amyloid plaque and starts impinging upon the membrane of the neuron, it damages the membrane. And this causes a whole chain of bad things, including, we found, the beginning of the tau pathology. And this is all in the mouse brain. What we were lucky is that one of our colleagues at Northwestern, Beth McNally, has been studying a protein that repairs membranes. It's called annexin A6, and I'll just call it A6. So what Beth found is that A6, when she studies, she was studying it in muscle from the angle of trying to treat muscular dystrophy, where membranes of muscles get damaged because of the muscular dystrophy. She's found that A6 actually heals the membrane of the muscle. So we thought, hey, let's give it a try in our mouse model of amyloid and see if we can heal the membrane of the neurons that's caused from the damage of amyloid. And sure enough, it is, there's hints that we are curing and healing the membrane and that it's reducing the tau pathology, at least the beginnings of the tau pathology. We still have a long way to go. We're writing this up in a manuscript that we want to get it published soon. And I'm very hopeful that, A6 actually might be a therapy. If we could get A6 into the brain, it might be able to heal the membrane damage caused by the amyloid, and that this, we would predict, would be a good process that would block the interaction between amyloid that promotes the tau pathology and the spread of the tau pathology.
And it would have to be earlier in someone's life, not to where it's just kind of too late till the damage is done. It's all getting back to that understanding earlier as the disease manifests.
Yeah, the key to to managing Alzheimer's is going to be prevention. To get people before there are symptoms and before there's a lot of damage done to the brain. That's what the A-beta targeted therapies are all about, like the A-beta immunotherapies and the BACE inhibitors. Those are probably the earliest. A6 is going to can maybe come a little bit later. And because it's right when the tau pathology begins to spread in the brain, that's when the memory symptoms start. So if we could get A6 into the brain right before that happens, then we might be able to prevent the memory problems.
Do you think there'll ever come a time in our understanding of the development of the brain, and when we don't talk about these diseases as diseases of the aged, but rather diseases of the process of aging? And what I mean by that is, we're looking earlier and earlier and earlier in someone's life to see when these things are triggered, whether it's happening at the cellular level or, something. If we find out that we really need to be testing people in their 20s and 30s, does this now take away the stigma, if you would, of, well, this is just a part of being old and just a part of being aged? It's really something that is the process of living that these things happen.
Yeah, that's a really good point. We don't yet understand the role of aging in Alzheimer's disease. We know that it's the primary risk factor for Alzheimer's disease. But, what is the mechanism of that? We don't yet understand. All we know is that we know a couple things. First of all, kids never get Alzheimer's disease. There are severe early onset forms of Alzheimer's that are genetic, and people have been known with some genetic forms to get Alzheimer's in their 20s. And so it can occur in early adulthood, right? If you have the genetic mutations that cause those aggressive forms. But in normal sporadic forms that do not have these mutations, there's something else going on that we don't yet understand that has to do with aging. And my own feeling is it's probably the brain ages and the vascular system ages, that there's less of a clearance of amyloid out of the brain, which there is a normal pathway. Everybody makes amyloid from birth, but their brains are able to clear it out. But as we age, I think that process gets reduced, and then it begins to accumulate. Maybe, as early, I guess it could start as early as 40, 50. It seems that it takes at least 2 or 3 decades for the amyloid to build up. So maybe, it is a process of just living and that as we age, things change, right? But we need to understand what are those changes so that we can circumvent them. One thing that seems to be true from the epidemiology is that exercise is important for delaying the onset or preventing Alzheimer's disease. It's one of the strongest lifestyle factors that is associated with prevention of Alzheimer's disease. And that the idea there is that it's mainly cardiovascular, that, if you have a good cardiovascular system, you can flush that amyloid out of the brain through the vasculature. But, I think in terms of society, we do have stigmas about aging. There's ageism all over the place. It's cool to be young and it's not cool to be old, which I think that's got to change because...
Yeah, I do too. And I think there's a lot of, we just don't do this when we're our age, right? We just don't play. We just don't put ourselves at risk of falling. And back to your, need for exercise. Well, you got to get out there and do things and run and jump and play if you're going to have a strong heart to have a strong brain. But so that, but you're looking at it. I think the superagers is super interesting. It's because the way I understand it, that's really you're studying these people over a lot of decades, and trying to understand, and then afterwards, after they pass, to try to even understand. So that's really interesting. What do you wish people knew about what you do and what your researchers do that they probably don't understand? Because, these diseases affect all of us. We all have somebody in our family, or we have friends, we know of people. What do you wish they knew about what you do?
Wow. Well, that's a good question. I think what I would like them to know is that we're all working tirelessly to understand the diseases that we study. And with that knowledge, if we can get at the underpinnings of these diseases, particularly at the molecular level, then we'll have a handle on, how we might be able to stop them, in a more of a classical drug development sort of way. But, we also have to be cognizant that there are non-pharmacological approaches that we can bring to bear on these dementias. For example, in the Mesulam Center, we have a program for the people with primary progressive aphasia. It's more of a speech therapy program because, their language is affected, their speech is affected, and, we've had grant money to be able to develop this program to help those people manage their primary progressive aphasia. And so, that's been very successful. So we want to approach these dementias from a multidisciplinary manner, where we're looking, not only at the genetics and at the molecular and cellular level, but also at the neuroanatomical level and all the way up to the clinical manifestations of the syndrome and the societal implications. So one of our mottos is from cells to social work, so we want to span the entire range of these dementias and superaging to understand, what's going on in their brains to produce their supercognition. And so I really would love people to understand that. Maybe if they were to understand that, they could be excited about partnering with us and helping us achieve our mission, which is to, I say, making Alzheimer's disease just a memory, right? And we need our participants, our study participants are the real heroes of the story. They are giving altruistically of their of their lives and eventually their brains to our research. And we couldn't do this work without them. So we are incredibly thankful and grateful to them for, their donations of essentially themselves. And they're true partners. We love to speak to people, to the public, getting the word out about what we do and why it's important, and get them involved as partners with us to solve these terrible neurodegenerative disorders.
Yeah. Well, just to make a small plug for the Karen Toffler Charitable Trust, we're very excited to partner with you and some of the scholars that we've given grants to, because these are early career researchers that are trying to create their labs, build their career, and just their they need support, financial support, and it doesn't take much. It doesn't take much to kind of launch that for them, and so they're pretty amazing. So as we kind of wrap up here, and I really appreciate your time and your insight here, and just the kind of the hope that you give of some of the breakthroughs. What is your greatest hope for the future? When you think out 5, 10, 15 years, what is your greatest hope here?
Well, I was saying before, I think my greatest hope is that we build our toolbox of therapies that really treat the underlying cause of these dementias, disease-modifying therapies. And we can't stop with just the 2 A-beta immunotherapies that we have approved now. We've got to have more. We've got to have different drugs that are targeting different aspects of the disease, not only the amyloid aspect, but also the tau aspect and the inflammation. And we know, for example, that there are about 75 different genetic risk factor genes that are out there. And so I kind of look at Alzheimer's disease in particular as maybe like the Mississippi River. So you have all these smaller rivers kind of trickling into the big muddy, which is the main pathway. But they all end up in New Orleans, right? With the same pathology. And so there are multiple beginnings, but at some point they all converge. And so we need to maybe design drugs against these different pathways. And then once we have this armamentarium of drugs, then we'd be able to have a precision medicine or a personalized medicine approach to an individual's Alzheimer's disease. So, what I can see in the future is people being, given a genetic test, see what risk factor genes they have, a blood test to see what, biomarkers are in their blood that maybe indicate pathology in the brain. Maybe everybody will get a baseline when they're 20 years old. And then every 10 years later, you'll get another blood test. And once you start seeing the presence of the pathologies in the brain, then you go into your toolkit of drugs and you go, okay, I see you have this genetic risk factor and that blood biomarker, and this brain scan is indicating if you're on these 3 drugs, this cocktail of drugs will help prevent Alzheimer's disease. But that cocktail would be different in another individual with different genetic risk factors and maybe different biomarkers. So that's where the future of Alzheimer's disease and dementia treatment will be. It's a true precision medicine approach, but we need more drugs. We need more targets, more drugs that are targeting those targets. We need new biomarkers, particularly blood biomarkers, that will tell us at what stage and what type of dementia these folks have. But I'm really hopeful. I think, and again, it's all about prevention. I think that once someone has memory symptoms, there's significant damage to the brain, and you're not going to reverse that. You're not going to bring back those dead brain cells, unfortunately. It's like heart disease. Heart disease, the example I give are the statin drugs. Statin drugs were developed initially from the finding that there were these rare genetic forms of hypercholesterolemia that led into the cholesterol pathway, and then the identification of the rate-limiting step for cholesterol synthesis. Statin drugs target that enzyme. And now, we have, serum cholesterol as your biomarker, and we can prevent heart disease to a great extent. And I see Alzheimer's going down that road eventually. But we still have a ways to go. Like I said, we need more drugs, more biomarkers, and early detection is going to be key.
Yeah, I love the visual of the Mississippi River with all the little feeders.
Yeah.
To reach that vision, do we need new kind of science too to be able to, more collaborative, to be able to see what all these feeders are for that individual? Because, science can be very stovepipe sometimes. Will we need to have new kind of capability, new collaborations to be able to see all the feeders that get into the big muddy?
Well, that's a great question. And certainly technology drives all advances in science. And we've seen, I started in the Alzheimer field in 1996, and I've seen tremendous advances technologically that have really advanced our understanding of, the natural history of Alzheimer's disease and the molecular mechanisms that we can now design drugs against. So technology really, really pushes the fields, any scientific field, forward. So I'm looking forward to new scientific developments, new technological developments. We're moving, I can see the move toward more collaborative science rather than small entrepreneurial, PI-driven approaches. I see maybe more team-based research on a larger scale. That's a little bit harder because us scientists have egos, and we like, to drive our own ideas, right? And it's one of my big passions. I love creativity, right? And I think every scientist loves that. And to be able to, have an idea and then test it in an experimental system. And, most of my ideas fail, but the ones that work are really exciting, and you get a real high out of it. So you might have to give that up a little bit if you're doing more team science. Companies do that really well, team science. And so maybe it's going to take partnerships between industry and academia and the NIH and foundations like the Alzheimer Association or the Cure Alzheimer's Fund, which I'm associated with. And in fact, these prevention trials that are going on right now are a collaboration, a consortium of companies, NIH, academicians and clinicians, and foundations. And I think, because these are huge trials, thousands of participants that are on this drug for 4 years, right? So it's a huge undertaking, very expensive. And, not even one company, even a rich company, can't really take that on. So these sorts of collaborations are really key, I think.
Well, they're so, these are hard problems. These are hard problems that you guys are trying to solve. And so, again, I really appreciate your time. Is there anything that I didn't ask that you wish I would have asked to get your message out there, or did we cover everything?
Well, you've done a really outstanding job of covering almost everything, and I can't at the moment think of much other than, again, to thank our our study participants. You know, like I said before, they're the true heroes. And we would not be able to make progress in any of these terrible disorders without them. And I can't say enough about them. You know,
and you need more. You need more.
We need more.
Yeah.
We're always trying to recruit new participants into our studies.
If there's people that want to just volunteer, do they just call? Like, how do they become a partner?
Well, the first step would be to go to our website. We have a website for the Mesulam Center for Cognitive Neurology and Alzheimer's Disease, part of the Northwestern website. And that might be the first step. And then they could kind of leaf through. We have different pages and pages that are describing some of the clinical trials that we're involved with, some of the studies, either pharmacological or non-pharmacological studies that are going on. And if there's something that piques their interest, they should, there's a phone number there. There's an email address. You can just, shoot us an email, and we can get involved in signing you up for something. So, we're excited to have all comers, and that's really the way we can partner with the community and really make advances.
Yeah, that's great. Well, thank you again. I know how busy you are, but making this time to share your thoughts and share your passion and just the effort. I appreciate you.
Well, thank you. It's been a real pleasure. I've really enjoyed this, and I hope the listeners out there get something out of it.
No, I think they will. Thank you.
You're very welcome. Thank you.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.